There are two groups that provide support to each of the groups in the Genome Sequencing Center. Those groups are Media Core and Materials Core. In Media Core, they prepare all of our liquid growth media as well as pour all of our agar plates. In Materials Core, they provide solutions for every single step in the process. In Media Core, they prepare over 10,000 plates each week and over 2,000 liters of liquid growth media. They also sterilize all of our glassware.
Why is it necessary to sterilize the glassware?
We have to sterilize the glassware because we reuse it. By sterilizing the glassware, it ensures that we get rid of all contaminants. If we had contamination, it might make us think that DNA from one organism belonged to another organism, and that would be a huge mistake. Okay, we're on our way to Mapping Core. As you know, a genome is all the DNA in an organism. This is way too much to sequence at once. The human genome itself is over 3 billion base pairs long. So, in Mapping Core, we receive a genome that has been broken up into pieces that are approximately 200,000 base pairs long. Each one of those pieces has been inserted into its own cloning vector, creating a huge bacterial artificial chromosome, called a BAC for short.
So you use bacteria to hold DNA from other organisms?
Exactly. And this trick allows us to prepare large amounts of a specific piece of DNA by starting with a single bacterial cell containing one BAC and letting that cell grow and divide. When we receive a genome in Mapping Core, we are actually receiving many individual bacterial cell lines, and each cell line carries one of the pieces of the genome in a BAC.
So if the genome is fragmented, how do you know which piece is which?
Great question. The answer is we don't know. The process that fragments the genome is random. So although a total collection of BACs contains all of the different genomic DNA fragments, we don't know in what order they fit together to make the whole genome. Here in Mapping Core, we put the fragments in order. We use restriction enzymes to cut the BACs at specific sequences, and then we separate the digested DNA with gel electrophoresis. This gives us fingerprints, or gel banding patterns, of the BAC DNA. Those BACs with similar banding patterns contain overlapping segments of the genome. Knowing where the fragments overlap allows us to construct a map of the fragments in the correct order.
So Mapping Core makes maps of genomic fragmented DNA?
And now that you know the order, you send all the BACs on to be sequenced.
No, that would be way too expensive. It's more efficient for the technicians to use a computer program. This program compares the overlaps and chooses the smallest set of BAC clones possible to cover the whole genome, kind of like stepping stones. We call this set the Golden Path, and it is this set of BACs that are sent through our production pipeline.
I'm guessing this is where the computer barcoding system comes in handy.
Exactly. It's very important to keep track of which BACs are sent on and how the DNA sequences they carry fit together. The first stop in the production pipeline is Library Core. Let's go there now.